and it's a pleasure. My first visit to Manchester City, Manchester University, Manchester in general. And I'm on an extended sabbatical in Cambridge. So if anybody is interested in continuing to collaborate and speak and discuss, I'm at least until this summer in Cambridge and possibly also for the next year. And I will tell you work that we've been doing. I'm a modeling person. I'm a theoretical physicist by my training, and we are working on different problems coming from biology. Cell migration is one of them, which is the one I will speak about today, but there's other problems to do with collector things like that. So if that's what you're modeling, you can approach me about that too. Why doesn't it work now? Oops. Okay. Yeah. You see? Okay. So cell migration is, of course, can we close at least just the lights in front of the screen? Just this one there.
I think it's got down.
Okay, brilliant. Okay. So cell migration is, of course, a very complicated biological problem. It's made easier if you constrain the cells to move in a pseudo one-dimensional way by, for example, making them move along lines that are printed on the surface, such that only along these lines the cells can actually grab the surface and adhere to the surface, and everywhere else they can't. You can do it along straight lines like it's shown here, and you can do them on hexagons, which I think you can make it that these are moving along hexagons. And of course, the motivation is that, first of all, you simplify the geometry of the problem from 2 or even 3-dimensional problems of cells moving in 3 dimensions or even in 2 dimensions to a problem of motion of this object in 1 dimension, which is easier to model and to think about. The motivation from biology, you can say, okay, this is completely artificial. The motivation from biology is that cells often do move along 1-dimensional tracks. For example, this is a brain slice from a mouse, and these green cells are cancer cells, and they are moving along very thin capillaries of blood. You can see a cell coming along here. There's a junction here. It moves to the side where to go, and finally it went to the right. So such pseudo one-dimensional motion is something that you also observe in the biological context. Yeah, long time ago, people were playing with these 1-dimensional tracks, and they did this cell race, and they found that there is a general correlation, although it's actually very noisy here because they mixed here very many different types of cells, between the persistence of the cell, how long does it continue along the same direction before it changes direction or stops, and the speed. And there seems to be like a general trend where speedier cells are more persistent. And that's where we started this modeling problem. The work was done together with Raphaël Vauturier from Paris, and Jonathan was a PhD student of mine at the time. And just a little bit before I show you the model is just a little bit of biology. It's inescapable when you're talking about biological systems like cells. So a motile cells can move with different strategies, but let's say the kind of motile cell we'll be thinking about today is a kind of cell which is propelled forward by this mechanism where it is continuously producing these filaments in the front, and these filaments that are building in the front are pushing the membrane of the cell forward in the front, and then the back has basically just to detach so that the membrane, which is this black contour around the whole cell, which is an elastic object which relates what's happening in the front to the back, is able to move forward with the pushing force due to these filaments that are being constantly built in the front. You can even see sort of wind or some flow coming from the front of the cell backwards. And the reason is that these filaments, as they form, they're pushing the membrane forward, but they're pushing also their own membrane and their own filaments backwards. So in the cell frame, you constantly have a flow of these filaments from the front to the back. So what is happening to these filaments at the back? They basically undergo disassembly, and there's a continuous cycling of these proteins from building them new in the front to push the membrane forward and disassembling them in the back. So it's a very, very cartoonish, simple-minded description, what I've just said, of the way the cell is able to propel itself. You also need to adhere to some substrate in order to move forward. If the cell is not able to adhere to the substrate, if it's like a slippery substrate, the cell is not able to move forward. So there is an additional issue of friction that has to form between the cell and these flowing mass of filaments in order to convert this into effective motion forward. Just to let you know that this recycling process of building these filaments and disassembly is constantly consuming energy. So this process on the face of it doesn't have a molecular motor that you usually associate with the conversion of chemical energy into motion. I'm sure many of you have seen all these beautiful movies with these molecular machines that are able to walk almost like humans with two legs. So this is not using molecular motors, but there's still a continuous consumption of the energy, of the chemical energy of the cell in the form of conversion of ATP to ADP, okay? I'm not going into the details of how this is done, but just to get you to understand that this is not a process or chemical reaction which is under equilibrium. It's an out of equilibrium process, continuously dissipating energy and converting chemical energy into a mechanical motion. That's all I'm going to say about how cells are moving forward. And now we go into the mathematical model. So the simplest model we could think of a long time ago, as I said, with Raphael and Couturier already, it's 10 years ago, was basically to think about the cell, although it's drawn here with the nucleus and everything, as just a one-dimensional object, where the filaments, for the moment, for simplicity, think of them as only being polymerizing at the front, and therefore, as they are pushing the membrane forward, they are pushing themselves backward. So if you go to the cell reference frame, there's a constant flow of these filaments backwards. Yes, they disassemble here. Let's forget about this process for the moment. Now assume that there is an additional molecule that can continuously hop on and off to this conveyor belt of these filaments. And when it's off the conveyor belt, it's just performing simple diffusion. When it's on the conveyor belt, it's moving with it at a constant speed v. So that's a simple advection diffusion problem. And if you solve it at steady state, you get an exponential distribution of the concentration of this molecule. I didn't say which molecule it is and what does it do, but as function of position along the length between 0 and the cell length L, you get this exponential behavior, with C tot being the total amount of this molecule integrated along the entire length, assuming that that's a constant, that's a given. There are closed boundary conditions. And the point is that this is an exponential which depends on the speed at which there is the advection, and of course also on the diffusion coefficient. So if the speed of the advection is 0, then this chemical is flat, it's the same everywhere. And as this speed increases, it's more and more swept to the back, okay? Now, the crucial point in order to make this into a model of how cells spontaneously polarize and begin to move is to complete the feedback loop by saying very ad hocly that the local speed at which the actin is polymerizing is itself dependent on the local concentration of this chemical. By local, I mean just 2 points, the front and the back. So our cell will be a one-dimensional object, and the local polymerization speed in the front and in the back will depend on the local concentration of this chemical, with some prefactor and some saturation concentration. So now you can see that the net flow of this protein, this molecule within the cell, is the difference between the flow that starts from the front backwards minus the flow that starts in the back and moves forward. So there's a competition between these two local sources of flow. And now you see that you have an implicit equation that you have to solve self-consistently for this resulting flow, because the flow, the net flow, will be the difference between these two local flows, but each one of them depends on the local concentration at one end and on the other end, but the concentration itself depends on the flow through these exponentials. Now, because of these exponentials, there is no analytic solution for this equation unless you expand it in Taylor series for very small v in order to find the behavior near the transition. But you can see from this structure that for, let's say, one of the parameters that we have in the model, which is, for example, the length, if the length is too short, then even if you start off with a velocity, a finite velocity, and very few of these molecules in the front and a lot of them in the back, it will not self-sustain. The concentration will always go back to being uniform and the velocity to zero. Now, the assumption here is that this chemical is acting like a local inhibitor of the polymerization. So the more you sweep it away to the back, you're inhibiting the back and further helping the front polymerize even more. So by starting this small asymmetry above a critical length, you have a runaway positive feedback, and you now have new solutions which are either a positive velocity or a negative velocity. Of course, going right or left is equally probable. And now there is a finite solution to this equation. The zero solution is always a solution, but it becomes unstable, and the two stable solutions are with a finite speed. Is that clear? Is that at least the feedback is clear? The faster is the flow, the more I'm sweeping this inhibitor from the front, so the front can even polymerize even stronger, and the back is inhibited, so it contributes less of a flow in the opposite direction, and therefore I increase my flow even further. I sweep even more the concentration, and this positive feedback can polarize the cell either going to the right or going to the left. So we have a model for spontaneous cell polarization. It needs some critical length, or it needs some critical value of this coupling parameter beta, for example, which has units of velocity. Now, so this was 10 years ago. Then 5 years ago, we extended that model by allowing our cell to be flexible. I won't go into the details, but now the cell length is calculated self-consistently by a balance of the forces that act on the front and the back. And the assumption is that all the cell mechanics are lumped into a simple spring. And that includes also if there is any contractility in this cell. You just crank up high contractility, will be like a very high spring constant in that sense. And now you see that you get the same general behavior, but slightly different. For example, you can drop the cell on your track, let it begin to elongate. As it is elongating equally in both directions, the local polymerization speed at both ends is equal, because this concentration profile is completely uniform, until a critical length is reached where spontaneously one is winning against the other. So the front is shooting up in its polymerization, becoming very strong, the leading edge of the cell. The back is inhibited, it goes down, and it becomes the trailing end of the cell. And that's the spontaneous bifurcation that signifies that the cell is not elongating symmetrically, but has decided to go right or left, of course, in this case, equally likely. And this is illustrated here. If the cell was dropped on your very long adhesive stripe, it elongates uniformly, but in this case, it doesn't reach the dashed line, which is the critical length in order to polarize. So it just stays sitting there forever, and many cells are like that. Or, as in this case, it actually does reach this critical length here, and now it polarizes. And now you see that there's a very big difference between how the front and the back behave. And that's nothing to do with the model of the polarization. That has to do with how we model adhesion. And we model the adhesion with what is called slip bonds. So you make bonds to the surface between the cell and the surface, but as one bond is detached, because the cell is moving, let's say, into the right, so these bonds are stretched and randomly they can detach. The off rate, the detachment rate, depends on the local load that is shared by the springs. And therefore, the more you're detaching, the same load is now shared by less and less springs, which gives rise to this runaway detachment, like an avalanche of detachments, which causes this huge and rapid detachment event where the back slips forward, and then there's again this slip, while the front is moving rather smoothly. And that's observed in many cell types. What also we have to remember in this process of slip is sometimes you can actually make the slip all the way back to below the length at which you are polarized. And when that happens, you lose polarity. So you undergo now elongation, which goes, which happens in the back and in the front equally, at least for a little while, until again, you're longer than the critical length, you polarize, and you can continue. And this looks like stick slip, but you distinctly have this event with polarity, and you actually elongate equally in both directions. And people see that in experiments. Yeah, we could compare to many anecdotal comparisons from experiment, doesn't really matter. But now we have the tool to approach the network, which is the title of my talk, so let's go there. So before we get to the network, the simplest element of a network is a junction. So I showed you a model where we can describe how cells move along one-dimensional lines, elongate, polarize, and commence moving right or left, stick slip motion, or even smooth motion. Both of these things happen in our model. But now we want a network. So the next element we have to introduce is a junction. So the simplest junction one can think of is a Y-junction. So 120 degrees, part of a hexagonal network. In fact, it is part of a network. You see another cell creeping in here. And now we want to basically take the same model that I showed you before, right? Before I showed you a model where we have just a one-dimensional cell. Now imagine this cell arriving at the junction and beginning to two new arms from that point onwards, right? The leading edge reached this junction, and now we assume that after reaching the junction, you initially start off growing two new arms, okay, and so on. Okay, I hope people see me on Zoom. Is that nobody's complaining? Okay, so yeah, so first of all, just to show you the kind of experiment, this is with high density of cells. We don't want that yet. We want to concentrate on a single cell. It's not easy if you have so many cells. You don't want cells to interact. Actually, you see that sometimes cells even move in trains. That's something we want to deal with in the future. The basic equations are the same as I showed you before. These are the people that were involved, and Pablo and Johan in Hamburg, and Neil and Pascal and Michele in Milano, and Giai and Jonathan in my group. And basically the idea is that now instead of just two ends that are competing for who will become the leading edge by sweeping this chemical, this inhibitor, all the way and inhibit the other competitor, now you have three competitors, okay? So that's basically the whole thing. But if before we had an easy time of calculating the concentration profile simply by having a one-dimensional diffusion advection problem, now we have a one-dimensional diffusion advection problem along three segments with a continuity condition in the middle. So we have three one-dimensional segments, each along each we solve this exponential profile. Now, how do we calculate the local advections? The idea is the following. It's shown here. If there is a certain speed or flow or flux of polymerization that starts from this arm, from this end, we assume that it is split equally, half and half, into the two remaining arms. The same goes from here. So this polymerization is contributing fully along this segment, but only half and half along the remaining segment. In the same way is calculated for all the others. So that's actually what is shown here. So the net flow that you have within a segment is fully the amount of flow that is coming from that segment minus half of the flows that are emanating from the other two segments that go into that segment. And with that condition, simple condition, we can calculate now the new profile of the inhibitor and introduce this competition between them. Is that clear? It's the simplest assumption that one could assume in this under this circumstance. That still conserves this flow. Okay, assuming that you agree or don't disagree enough to stop me, we can actually now use this model to calculate the dynamics of this cell on this Y-junction. So for example, what we find is that you can now drop your cell on the junction, on the junction itself, and let it spread equally in all directions. Again, if your activity level of the cell beta is less than some critical value, your total length, which is the sum of the three arms, will be less than some critical value, and therefore you'll just stay there with three arms equally, not moving anywhere. If you are with activity larger than the critical value, your length grows above some critical length, which we can calculate analytically, and then you see that there is now a bifurcation. One of the arms is losing, but the other two are still of almost equal activity. Not exactly, because you see that the red, for example, will now drop down, and the blue will continue to climb all the way up. And indeed, the blue is the one along which the cell leaves the junction. So the cell leaves the junction and becomes motile along one of the arms, doesn't matter which one, it's the blue one. And you see another interesting observation here. While the green was the loser from the beginning, well, almost from the beginning, from the time that the length crossed the critical length, the red, which was the second runner-up, when it came its time to lose, it didn't just join the green one at the bottom of the pile. Actually, it made the green one recover in its activity almost fully. And you can see also that the green in the length is actually lengthening now here. You see it's climbing up. So although this arm was already very small and short and getting shorter because the others were lengthening, and just because of elasticity, it was getting shorter. Now it is actually elongating. You see it a little bit in the length, but you see it even more dramatically in the local activity at the tips, which is this local polymerization that happens at the tips. And the reason is that the length is, if you think about it, it's like an integral over these forces over time. So it's a much slower and weaker effect, while the jump here in the activity are much more dramatic. So this immediately caught our attention, and we thought to ourselves, okay, what is happening here is actually far from trivial. Not only we have like a hierarchy of who's losing slowly, first one is losing, and then the other one is losing, and then just leave along the direction of the winner. As the second one is losing, the other losing one, the one that lost first, is actually recovering. So you have some oscillation here, because you see there's even another oscillation where after some time the green collapses and the red resurrects, not fully, and then of course it finishes. And we can understand the mathematics of this seesaw oscillation. It's not a traditional limit cycle behavior, because the system here is not repeating. It's not in periodic orbit. It's actually moving one directionally from being very small to leaving the junction. But we call it seesaw oscillation because it's like when one goes up, the other one goes down. And we can understand why that actually happens from the nature of these solutions. But there's no, I don't think it's super intuitive. Just to tell you a few words about what is the level of activity that we work with. So as function of the level of activity, this parameter beta of the cell, and you can also look at the elasticity, which determines how large is the cell. If the activity is too low, let's say for a given elasticity of the cell, then the cells are trapped. They are like this. It just grows three equal arms, and they cannot start moving. you remember that also for a one-dimensional line, we said that if the activity is too weak, they just elongate equally and stay without polarizing. Well, apparently, you need a stronger activity in order to get out of this trap. This is a stronger trap than just being trapped on a line. Then cells can actually move and escape the junction, like shown here. But surprisingly, when the activity is too large, then there is another kind of behavior, which we call the slow process, where a cell arrives at the junction, two new arms form super quickly because there's such a high activity of the cell. The back, which was here, basically shrinks to zero, but now these two very elongated arms are stuck in this indecision for a relatively long time, and that's why we called it a slow process until this is resolved eventually by some noise. So, but before that, let's just go a little bit on look a little bit on these oscillations. We're very excited by these oscillations, and we asked if we can see them in experiments. So this is, for example, the glioma cells, these brain cancer cells. The movie, which you show here, shows them coming to a street, to a junction. It went first to the left, but eventually it left to the right. And if you see the length of the arms, this is the back shrinks, then goes up, and then eventually goes down. The green was growing, but eventually dying away, and eventually it left along the blue arm. But we wanted to see this activity at the tips. So we could not actually measure actin polymerization rates at the tips, but what we could is define the size of this lamellipodia, or this protrusion that locally the cells flare out when there's a lot of actin polymerization, these filaments that polymerize in the front in order to pull this protrusion, this arm forward. So we could use that area at the tip of these three arms as a proxy for what is the local activity of the actin. And there's a little bit of an out of phase between the green and the blue, the green and the red, which are the two losing arms, which is what we expect. It's not so convincing maybe here, but maybe this is a nicer example. That comes from endothelial cells. And you see many events where the red and green really oscillate out of phase. I think from here there's one oscillation, second, something like 3 oscillations, where it's clear that they are oscillating out of phase, the two losing arms, which are the green and the red, and that's the simulation, which has roughly the same characteristics as what we see here from the length point of view, and also from this number of oscillations that are there. Yeah, you can also see it even in I mean, at least this structure of the changes in the length in this glioma cell moving these capillaries in the mouse brain slice. You see that it grows first to the left, then to the right, and eventually it moves out from the right. So again, there's a little bit of an oscillation here, which again might indicate that that's the kind of oscillation that we see. This very slow phase that I told you, with the two arms getting extended, and then the back is completely at zero, and these two arms are almost at the same length until just at the end there is a collapse of one of them, and only the blue is the winner. And you can actually find experiments that look similar, especially with cells that are very active, like the glioma cells. You see that now it will reach this condition. You see the length of the back collapse to zero, and the two lengths of the two arms are almost identical, just like we have in the model, until they collapse. But they collapse by detaching from the node here, because they make a bridge over this non-adhesive region, which of course in the model we can't allow that. I mean, in principle. So it's not exactly resolved in the same way at the end, just because of this ability of the cell to detach from this junction and just bridge. But before that point, it looks very similar, we think. And you can look at why we call it a slow process, because under these conditions of very high value of this beta, of this activity, where we get these processes appearing, you can see that there is still a chance that the cell will actually go through the junction very fast. That's this bar here on the histogram plot of the escape times. But then there's a huge distribution of these escape times of this slow process, which extend by an order of magnitude into slower times compared to the distribution which is hidden here, which you can see when you zoom in on these fast processes. If you look at the glioma cells, we think that it does show signs of that because they are moving very fast, and there is a distinct sort of secondary peak here of events which are much, much slower than the mean of the glioma, while the UVEC show something which is more like a uniform distribution. If you look at the model, you have another control parameter which we haven't spoken about yet. I've not told you anything about it, and that's the noise. So everywhere I told you that at each of these tips of these arms, there is a local polymerization, which we calculate from the local concentration, and the concentration is affected by the flow. So everything is calculated self-consistently as time goes by. But we can also add noise into this process by saying that the local polymerization activity also has a noise term. So it's calculated from this inhibition plus some noise term. And if this noise term is very small, for example, you can see that the distribution of escape time, the time it takes the cell to go through the junction, is nicely peaked, and the probability of where will it go is 50-50 between the two new paths that are open to it, okay? As you increase the noise from 0.4 to 0.8, there is still the peak at values which correspond to this kind of process, which is the process of just going straight and either to the right or the left. So you see the probability is the same 50-50 of the green and the red. But then there's another peak of slower processes where there's now a very high probability of actually going back along the old path. So with larger noise, you have a probability of actually reversing at the junction. And if you think that's crazy, there's actually an example here where the cell arrives at the junction. These are the two new paths, but instead of going along them, it actually prefers to go back. And that is exactly what we predict that can happen as the noise is cranked up in the model. If the noise is super long, what happens is that basically the escape time becomes very long, and there's now a third and a third and a third of probability of going into each of the three directions, which means that essentially the noise is so large that after moving forward and getting to the junction, the cell has completely forgotten where it came from, and it now spends so much time on the junction that it's 1/3, 1/3, and 1/3 to get out of the junction in any of the three directions. And there's even events where the noise is so large that you can spend a very long time on the junction, and that is shown here. This is an experiment where the cell arrived in the junction. It was moving fine along the 1D line, but once it reached the junction, it became trapped in this indecision, and you can see the length of the three arms. And this is a simulation from the region here, where the escape time is very, very long. You have almost eventually, if you do get out, you have a 1/3, 1/3, 1/3 probability of going in each three direction because you forgot completely the direction you came from. Yeah. Yeah.
Do you take into account that the tracks have changed? Like, is it what along it?
We didn't. And there's new work from Benoit Ledoux's lab in Paris, which shows that in fact cells do leave a footprint, and it affects them. Yeah, yeah. It affects them in the sense, how does it affect them? It affects them in the sense that they like better to go along on paths on which they've already walked on. So you're right that in that sense, if it needs to break new ground here, it'll be easier to go back along the old one. But we did find many events where it didn't seem to care. So I don't know if that's a big effect in our data. So these experiments are done where this is a surface, I don't remember which, but it's already been treated such that here it's been covered by some polymer that makes the cell unable to stick to it. And these are the lines of the hexagonal pattern, which are made of collagen or some other ECM, which is easy to stick to from the. Yes, yes. Okay. Now finally we get to the networks. Yes. So after we have a model that can describe a cell moving and elongating and polarizing spontaneously in 1D, and also now negotiating a junction, the next logical step is to deal with a network. Now for that, you recognize already some of the people here. I think this is a new face, Javier. We need now to consider a cell that might span more than one node on the junction. So first of all, this is how experiments look like. This is a macrophage. I think you can just about see that it's spanning several such hexagonal nodes. And you can see also in the simulation, sorry, you can see that now this object can span more than one junction. So it has clearly more than 3 arms. So it becomes more complicated, but it's essentially the same model as I showed you before. Okay? And this was just to motivate you, to show you that there are these immune cells that are constantly navigating in your body between the other cells, which you don't see them. They are sort of the holes, and that forces these immune cells to get these very highly branched shapes because they are constantly trying to squeeze in between the other cells. And that's what they do constantly in your body. You can see that, for example, in this image taken in vivo from the tail of a zebrafish larva. And this is, again, immune cells, which you see are highly bifurcated. That's the images. And that's a skeleton version of these images. And you can see them even in the fluorescence moving inside the tissue. Yeah, here I show you another example. This is again that are not moving very much, but I mean they're moving, but the cells are really not much, but they're clearly high branch, most like this hexagonal network of the cells that they are trying to squeeze around them, and they are constantly searching for pathogens and infection, and that's what keeps us alive. Yeah, and Nikolai was good to share with me actually this really nice movie, but now nobody on the web will see it. Okay, so only you see it. Sorry. Yeah. Oh, I can't even do that. Okay, I can't even do. Okay, sorry, I couldn't embed it in the PowerPoint, so I'll just go back to the PowerPoint. Okay, sorry. Sorry. That was my mistake. Does it work now? Can you see something? Yes. Okay. Okay. So the network model is essentially the same model as I showed you before. Oh, how do I get rid of you? Sorry. Yes. Okay. So it's essentially the same model that I showed you before, with the flow that starts from here being split into 2 at one junction, and then it gets split into another factor of 2 in the second junction. So the effect of the advection activity from here all the way down to here is of course diluted. And we considered shapes of cells up to spanning 4 nodes, just like this one. And still the competition is the same. Whoever wins tries to push this inhibitor all the way down as far as possible from it down to the losing end, let's say this one or this one. So that's still the competition. So okay. So it's the same rule as I said before. And now we can actually now study these migration of these objects on such a network. We can look at the number of junctions that this object is spanning as function of how active it is and the size d of the network. So this is the size of this hexagonal network. So not surprisingly, you see that when the network is very sparse, so its sides are very large, you at most span 1 or 2 junctions. And as you are making the size smaller and also increasing the activity, your cells become longer, and now you can span up to 4 junctions. So that's the time series of the number of junctions that this object was spanning: 2, 4, 3, 2, 2, and so on. And the total length of the cell is changing as well. So you can now look at this behavior. What you find is, for example, that as you compare these two examples that only differ by the size of the network, you can see that when the network is sparser, so the cell cannot span so many junctions and therefore necessarily have a smaller number of arms, it's moving with a slightly larger average center of mass speed. It's slightly higher, the orange, than the blue. But what's most important is that its maximal difference of this concentration of this protein, which is this inhibitor, between the front and the back, is much larger as compared to the case with more junctions, with more arms. So the more branched is the cell, the less it is able to produce a strong difference between front and back. And that is easy to understand intuitively because you have more points competing for who will become the leading edge. So with more competitors, you get more of a uniform distribution of this inhibitor, and you are able to less polarize it all in one direction, right? In 1D, it's the easiest because you have one competitor who won, one who lost, and the loser gets all of this inhibitor, and therefore the difference is maximum. So the more these cells are bifurcated, they are less persistent. And we see the effect of that also when looking at the large-scale diffusion of these objects on the network, and that's their mean square displacement. So first of all, the mean square displacement increases trivially with the activity. So the stronger is the activity pushing the cells, the faster are the cells. Trivially, the diffusion coefficient is increasing. And what we also see, if we look at the color map here, which is the diffusion coefficient, as we make the network denser, so between looking at the sparse network where D is large to a dense network where D is small, the diffusion coefficient is decreasing. So you see, by going from here to here, I actually cross some of these contours and I go down to a lighter value. So I'm going down. There is some slope here for these contours. So in general, going down and into a denser network, even for the same activity of the cell, I'm able to diffuse less efficiently. And again, that has to do with the fact that I'm more bifurcated and I'm less persistent. And you can see that in a movie, that the more dense is D equals 3 and the less dense is D equals 3.5. And you can quickly see that the D 3.5 is actually moved out much more because it is more persistent than the D equals 3. It's not a big difference in the size of this hexagonal network, but it already forces this one to have, on average, more branches and therefore be less persistent. Okay, so that's already something that the model is telling you. And again, is that what happens in experiments? I don't know, because they have not done large-scale diffusion experiments on these networks. So all I can compare with, that's an example of such a trajectory. Let me, yeah, you can now go into all kinds of details using these simulations. You can look at the distribution of the residency time on a junction. And for example, in this case, you find that I think it's a low noise and it's some activity level 7, whatever, and there's some size of this network. You find it, for example, the residency time on the on the junction depends if you spend just one junction, two junctions, or three junctions. And you find that, for example, for two junctions, you have a very long, a population of very long residency time. So it's a very stable configuration that that comes up in this kind of shape, which is more difficult for the cell to get out of and continue onwards with its diffusion. So you can go into these kind of details. Unfortunately, at the level of the experiments that we have today is very short trajectories. So all we could do is just compare this very short time dynamics of the length. So this is the experiment. So the total cell length you see also oscillates. This is the simulation. And now you plot the lengths of different arms, which you color by the many colors that appear here. It's a bit of a mess, but we try to convince that it's not such a unrealistic comparison with the model. If you look at something more quantitative, like again, the distribution of residency times on a junction, irrespective of how many junctions you cover, and if you separate it by the number of junctions that you cover, I don't know, again, you can try to do this more quantitatively. This is that I showed you previously in this. Now, of course, you see that the algorithm that picks up the tips is not perfect, but that's what we have. And that's the kind of analysis that this kind of algorithm allows us to do. This is just to show you that by comparing different cell types, we can say which cell corresponds to a higher or lower value of this parameter, or maybe we can even say that by this comparison, one cell type seems to be more noisy than the other. But again, it's not very quantitative, I would say, just a qualitative. By comparing the motion of macrophages, I think one nice comparison shows that the residency time over a two junction, which is objects like this, which span just two junctions, again, seem to be very stable in experiments, and they do have this longer tail also in the simulations compared to spanning just one junction or three junctions. So again, I think we can make some comparisons of this type. I will just finish by telling you the work we are doing now with Nir Gov in Cambridge, which is to use exactly the same kind of modeling approach to think about a specific problem from biology, not just moving randomly and aimlessly on a network, but actually moving towards a wound. So this is a zebrafish larva. Milka is making a laser wound, like here. And then you see that all these macrophages, which were rather dormant and pretty much uniformly distributed everywhere in the tissue, suddenly wake up, and all of them, boom, rush to the wound. And that's, of course, super important also for us, because this is the first line of defense. If these neutrophils don't rush to the wound, bacteria do rush into the wound, and we are dead. So it's really a matter of life or death, and the light here is very bad, but it shows you this neutrophil aimlessly moving around. Then there's the wound here. It stopped in its track, and now it's rushing to the wound like everybody else. And you see even in this neutrophil that it's quite highly branched. And again, for the same reason, all these immune cells are branched, because they need to move all in between this tissue. They don't have any other choice. If you just plate them on a glass slide, sometimes they don't look so branched, but this is not the usual condition. So I'll just leave you with this interesting result that we not published yet, and we looked at the ability of our project to discern a chemical signal. So think of this cell that I talked about all the time in our model, arriving from below, coming into this Y-junction, but along one of the arms, there's a tiny little chemokine source. And we modeled that by saying that the local activity of the actin that is pushing the front here is slightly larger than the beta zero, which is everywhere else. That's it. Now, what we find is that as we crank up the activity of the entire cell, the time it takes for this cell to cross the junction, I should have said here the time to cross the junction, is monotonously decreasing, at least up to here. What happens here, we'll talk in a second, if we have time. But what is more interesting is that when the cell was very slow and took a long time to pass the junction, you also find that the probability of taking the wrong decision, which means moving here, is low. It's about 20%. As it is moving faster and faster, the error is increasing. And that's a typical speed accuracy type of behavior that you're used to also from your own life, right? If I tell you to solve a problem in 5 seconds, or I give you 50 seconds, you get it right better with the 50 seconds. So speed accuracy trade-off is something you find everywhere in biology. Here we find it in this simple model. And until this point where there's a huge jump in errors, almost up to just flipping the coin 50-50. And what happens here, which is shown by this blue shaded region, is that suddenly there is an appearance of a probability of not only going in the wrong direction in here, but actually also of going in the wrong direction going back. That's the probability of going back, which was essentially 0 up to that point, and suddenly it goes up. Going back is also the wrong decision. We have to go to this. This is the source of the wound or infection or whatever. And that's the reason that there's this big jump. And actually it appears in this case because of above a certain speed, we begin to get these huge stick slip events that, remember, we talked in the beginning. And that means that as the stick slip event happens, it can happen sometimes right at the junction. And then this poor cell finds itself losing its polarization at the junction, which is a bad place to lose your polarization, because now you forgot where you were going and there's a chance you will actually go back. So I will just not have time to talk much about this chemotaxis problem, but I will just leave you with some open questions. One open question, which maybe the mathematicians here would like to be interested, is what happens in random network, because real cells are moving in something which is not perfectly hexagonal, and we've just started to think about how to quantify such random networks, but we haven't actually let our object move on them yet. So this is for the future, and thank you very much.